Good morning, class, or afternoon if you're watching this later in the day. This is the second of my videos, and I'm going to post some more. But before we move on to new content, I guess I need to go over some of the older stuff that we just covered before we went on this surprise break. We stopped with mitosis. You're supposed to be doing some reading, and you have a copy of the textbook, both virtually and a hard copy. And if you need to review mitosis and cell cycle, it is on pages 130 through 139 in the textbook. There's also pictures. If you're not one for reading much, there's also several YouTube videos, including this, that kind of recap those concepts. You've got to understand mitosis first before you can go into meiosis. And I will be doing meiosis later on. It may be a week or two before we get to it, but it will make very little sense until you understand how mitosis works. The end goal is different for both of those, but the terminology and a lot of what's going on is basically the same thing. All right, speaking of reading, let me go ahead and throw this in. The district is wanting all of our students to read, not only for school, but also for pleasure. Reading helps expand your vocabulary and implants new ideas into your head about the world around you and some different ways of seeing points of view, and also may teach you some history, some science, may help you with a lot of different things in the real world. Currently, I'm reading The Sixth Great Mass Extinction by Elizabeth Kolbert, and I haven't gotten very far in it because it is spring, so I'm trying to plant the garden and prepare it for the spring and summer harvest, but I'm going to try to continue on that book. Reading kind of slows down in the springtime because of all the things that need to be done outside, but I hope to sit down and read a few chapters every few days. Hopefully you too are reading. All right, so for mitosis, let me grab the whiteboard. All right, you may remember this from class. So mitosis, is a cycle. So there's not really a official beginning and end, but we like to start with G1 because that seems a little bit logical. In G1, G stands for growth or gap. The cell is growing, and this is actually majority of the life of most cells is in G1. I may have made it seem smaller than it really should be. It's here drawn on this little circle. It's only about 45% of the pie, but in most cases it may be over 90%. If we're talking about neurons inside of the brain or in the spinal cord or even down through the tissues, neurons don't go through cell cycle very fast. So those cells can stay in G1 for over 90% of their life. But some cells have a shorter G1, like for example the cells inside your nose. Those regenerate every few days, and theirs will be much shorter. So G1 is growth 1 of the cell. That's where the cell does most of its normal activities. It will do whatever its job is. If it is a neuron, it's sending messages. If it is a gastrointestinal cell, it is producing molecules to help break down and absorb food. If it is a muscle cell, it'll be contracting and releasing, and contracting again and releasing for whatever is needed. G1 is where a cell does its normal job. After G1, we go into S. S stands for synthesis. To synthesize is to create, like photosynthesis uses photons, light, to create sugars in plant cells. Well, synthesis here is it synthesizing DNA. If our cell has only one strand of DNA, like bread mold, then in synthesis it's going to take that one strand of DNA and copy it and make a second strand. We do not have one strand of DNA alone. We have 23 pairs. So those 23 pairs are going to become 46 pairs of chromosomes. After S, and that can take hours, maybe up to a day or two, to copy all the DNA. It's not a super fast process, but after that, the cell will go back into G2. G2, again, is growth. The cell is going to grow, and it's going to start copying all of its organelles. G can also, besides standing for growth, can stand for gap. A gap in manipulating the DNA. In S, we copy it. We're manipulating it. Then once we exit G2, we start manipulating the DNA again in mitosis of cell cycle. So G could be a gap in manipulating and changing the DNA. So we exit G2 and we go into the 4 phases of mitosis. Officially mitosis: prophase, metaphase, anaphase, and telophase. These 4 phases you'll need to know for meiosis. They're not too hard to remember. PMAT, P-M-A-T. Prophase, pro means before. So before we can manipulate the DNA, we need to do some things. We're going to break down the nuclear membrane, also called a nuclear envelope. We're going to send centrioles to the 2 poles. They serve as anchors to help pull the DNA apart. And we're going to create little ropes called spindle fibers. Those spindle fibers are going to pull the DNA to the 2 different sides. After prophase, we go into metaphase. Meta means after. After we've set everything up, we then can start moving things around and doing stuff. The DNA is going to get pulled to the middle of the cell, and they start lining up. Speaking of the DNA is going to look like these, like little X chromosomes. They're 2 sister chromatids, and our goal is to separate them into separate little strands. And that's going to happen in the next phase, which is anaphase. Ana means away. We're going to tear these 2 strands, called chromatids, apart. Excuse me. I was spreading hay in the garden yesterday, and I breathed in a lot of dust. So, anaphase separates these 2 from one another. They should be identical twins, and we want to separate them so each cell is going to have its own copy. After anaphase, we go into telophase. Telophase is where we start to put things back together. We're going to rebuild that nuclear envelope, or nuclear membrane, if you want to call it that, and we're going to repackage that DNA. The DNA in pro, meta, ana, and telophases was wound up in nice little packages around proteins called histones. And that's very convenient for moving the DNA, but what we need to do is unwind it, kind of like a garden hose. If you wind it up to move it, that makes it much easier to move it from point A to point B, but it's not super useful in using the garden hose when it's wound up. We need to unwind it. So in telophase, towards the end, we're going to start to unwind that DNA so it can then be read. After telophase, we go into our last piece of mitosis called cytokinesis, there in red. Cytokinesis is where we are breaking the cell into 2. Cyto refers to the cell, and then kinesis means division. So we're dividing the cell. I'll put this down for a moment. Cytokinesis looks a little bit different in different types of cells. If we lack a cell wall, like for example, animal cells do not have a cell wall, then the cell membrane can just start to bend on itself until it splits into 2. If there is a cell wall, then we can't bend that cell membrane easily. There's too many molecules there that are rigid, stiff, and don't bend freely. So what we'll need to do instead is build a new cell wall and new cell membrane down the middle of the cell. While it's under construction, it's called a cell plate. Once the cell plate is done, it'll then be called a cell wall. But we need a name for something that's under construction there. So cell plate is the construction name of the new cell wall. Then, after cytokinesis, the whole process starts all over again. We go back to G1. The cells will be much smaller because they just got divided into two. So they need to grow, hence going back into growth number one. Once it grows and does its normal job and prepares proteins and does its normal functions, then maybe a signal will tell it time to start to divide the DNA again, and it'll go into S, and the process continues around and around and around. Some cells do this very, very fast, like for example, I've already said the ones in your nose and your gastrointestinal tract. Then there's some that do this extremely slow, like I've said, the neurons in your brain or in your spinal cord. They will take years and years. Some of the neurons don't divide for every 30 or 40 years after you get out of a few months after being born. I hope this helps, this review. The next video that I'm going to post will be on cancer, and that's a topic that we really didn't get to go into in class. And I'll recap the little bit that we started with and then go into some new things that we have not. If this video was helpful, great. If not, then I'll also post some other videos from other sources like Crash Course and Amoeba Sisters to help you understand mitosis better. Again, this is a concept you gotta understand to understand meiosis. I hope you have a wonderful day. Use your textbook. Oh, right there. Weird angle. And then also remember to read something for fun. This is what I'm going to read for fun. Have a good day.